Hello, and welcome to this week's episode of I Came for Science, a show about sex and reproduction and inclusion in STEM. I'm your host, Laura Surratt. I'm a professor of biology, and I've been studying animal sex for the past 30 years. Please join me as we normalize talking about sex and what we know about it, as well as the journeys of the scientists who study it. In a recent episode, I spoke with Matt Dean and Dave Hoskin about all of the amazing penises in the animal world and how they evolved. In that episode, we also discussed how we know a lot less about female genitalia. Our guest today, Dara Orbach, and other researchers are working to fill that knowledge gap. Welcome, Dara. Thank you so much for joining us today.
Thanks so much for having me.
Dara is an associate professor of marine biology in the Department of Life Sciences at Texas A&M University at Corpus Christi. Dara, I want to start by sharing some stories about weird female genitalia. What are some of your favorites?
One example that I really like is the work of my postdoc advisor, Dr. Patricia Brennan, who's been foundational in the study of genital evolution. And what Patty found is that although most birds have a cloaca, so they don't have a penis, an intromittent organ, they don't have a vagina, waterfowl are different. And Patty
Can you define cloaca?
A cloaca is a central opening where you would have both the reproductive tract and also the digestive tract would all mix together in one area. So it doesn't have the structures you would find inside like a penis or a vagina. It's one giant area where feces and urine and sperm would all come together.
And both males and females have this.
Correct. And the way that birds mate is they do something called a cloacal kiss, where they touch their cloacas together, and that's how they transfer that sperm to one another. So they don't have a intromittent organ, so intro going in, which is another way to say penis, because not every animal has a penis per se. Like fish, they have a different name for them called a gonopodium. So what
in insects we call it adiagus.
Adiagus. I've been saying it wrong all this time. So what Patty found is that males have a penis which spirals in one direction, and females have a vagina which spirals in the complete opposite direction, which means they don't actually fit together. And if you think about this from an evolutionary perspective, most birds are monogamous. You have one male mating with one female. But what happens if you are a male who's unable to secure a female mate? You still are going to want to mate with the female to pass on those sperm and sire offspring. And so they engage in a behavior called forced extra-pair copulation, which does not sound particularly pleasurable from the female perspective at all. So you can imagine that if you are a female and your genitalia spirals in the opposite direction of the male's, it is not going to fit well together if the male is forcing himself on you. But if the female does want to mate with this male, she'll actually contract those vaginal muscles, and that will relax the shape of these spirals and allow the penis to penetrate very deep and the sperm to have the shortest distance to travel to fertilize the eggs.
That's so interesting.
It's this genital coevolution between males and females, and a really fun, I think, example showing how both anatomy and behavior work together as well.
Absolutely. There's something similar that happens in cucumber beetles, which is interesting. So it's not so much the shape of the genitalia, but males will take their adiagus and intromit into the female, and she'll tighten her muscles, and he won't be able to get his penis all the way in to where he needs to be to deliver sperm. And he starts tapping her with this copulatory courtship. And if he taps just the right way for just the amount of time, then she'll release the muscles and allow him to intromit. So it's very interesting.
I was thinking even about bed bugs and the idea of traumatic insemination, and that the male pierces her female inside her abdomen, but it's a particular spot where the cuticle is softer called the spermolage, and it's soft so the female heals faster. So you have this coevolution over time as well, where there's less damage to the female. And that's something I've sort of found with my research too. We found that marine mammals, particularly cetaceans, which is the group of whales, dolphins, and porpoises, have the most diversity in their vaginal morphology compared to any other vertebrate clade. So I'm at a stage in my life where when I get sent packages in the mail of different species, I can tell you what the species is just by looking at their vagina. And we know in insects, penis diversity is extreme, and you can tell species ID based on penises, but that's very uncommon to be able to do that with a vagina. So cetaceans, extraordinarily diverse. The penises are actually not as distinctive. There is variation across them. And I happen to have studied more vaginas than penises just because that was my initial research question. Now I'm looking more about how they fit together and looking at more penises.
Sure.
but the vaginas seem even more diverse compared to the penises.
That's
And
interesting.
really, they should co-evolve together. So if you're seeing
Mhm.
extreme complexity in one sex, you should see it in the other sex as well.
Mhm.
And so these cetaceans, these whales, dolphins, and porpoises, they have something called vaginal folds, which are protrusions of the vaginal wall into the vaginal lumen. And these were first described in the 1700s and not really thought about since then. And the thinking was that maybe this was a way to prevent the fetus from falling out of the vagina, which doesn't make a lot of sense if the
Does
fetus
not
is
make a lot
housed
of
in the uterine
sense.
horn and not in
No.
the vagina. And then
Yeah.
another thought, which was that perhaps these were a way to squeegee off the penis because it turns out that salt water is lethal to sperm. So maybe it gets
Squeegee.
all the water off.
I love that. Squeegee. Okay. Yes.
But if you think about it, most of these vaginal folds are pretty deep into the reproductive tract near the cervix. If the
Mhm.
goal was to keep salt water out, you would expect them to be right at the opening of the vagina, not at the complete opposite end over there. And
Right.
why would there be so much diversity if the goal was just to keep water out? And then
Mhm.
we also know that river dolphins, which don't live in a marine environment, also have these vaginal folds, whereas our other marine mammals like manatees and seals do not have any structures like these. So there's no convergent evolution. So this got me thinking that maybe these vaginal folds play some sort of role in sexual selection. And the whole way that I landed up in this field was really fortuitous that I used to be a bat biologist and study bat bioacoustics. So I've studied sex, obviously, but my master's was looking at alcohol. And most recently, I've been looking at drugs. So I've made my career in drug, sex, and alcohol and animals. But I
Okay.
used to study bat acoustics, and I knew I wanted to switch to marine mammals because I'm happiest when I'm by the ocean and really
Mhm.
want to spend my life looking at the water always.
Right.
And
Yeah.
so I went to a marine mammal conference, not knowing a single person there. There were 3,000 people there.
Wow.
And I was determined to find a PhD advisor. And I was going into random talks that I thought were interesting. And in one of them, this graduate student said that he had studied the species called the dusky dolphins in New Zealand. And it was one of the first studies ever describing mating behavior in any cetacean species, which is
Mhm.
shocking that in 2009 we know so little about mating behavior in general. And at the end of his talk, he said, "Oh, and they have these weird vaginal structures." And being a comparative biologist, I started to think across systems. And I had learned that spiders, I heard at a seminar that spiders, when they mate, the male taps on the female and it can force her to ejaculate sperm from a rival male. And so it
To
got me
eject the sperm.
thinking,
Yeah.
eject the sperm
Yeah.
ejection.
Yeah.
So maybe these vaginal folds or these vaginal structures in cetaceans were also a way to control paternity. And that's what led me down the path of my whole PhD, which was quite outside the box because other people have not really been looking at genital evolution in many species, especially not in our marine animals. It's very hard to observe them mating in the best of times because it can occur offshore and subsurface. And these animals don't necessarily die close to shore. They might strand in the middle of the ocean and sink down or be scavenged. So there hadn't been a lot of research. And I sort of fell into this field just by going to a conference and having this outrageous idea, which has been extremely fun and enlightening. And I'm so glad that I went to that conference and went down the journey I'm in now.
That's
And so one
awesome.
thing we found is that harbor porpoises are absolutely amazing for many reasons, but one is their mating. So the penis is 3/4 of the male's body length. It comes all the
What?
way to his chin.
Oh my gosh.
So these
So is
little
it sort it's like
porpoises,
tucked in normally or
like
no?
in general, because if you think about mating in the ocean, if you had anything external like a penis hanging out, there would be drag forces acting on it.
Right.
And our marine animals are extraordinarily streamlined. So their genitalia is internal. The penis is
Mhm.
kept inside the body, and just before mating, the male will avert his penis. And so these harbor porpoises, which are thought of as being shy animals that are relatively solitary, are anything but that when they're mating. The male approaches the female from far away. He comes at her from deep below and always on her left-hand side, and he leaps through the air when she's at the surface taking a breath. So he coordinates his timing that he can use the water like a wall and trap her against it. And he leaps through the air and he wraps his penis around her body while
Wow.
midair into her vagina at such force that it pushes her out of the water. It is absolutely
What?
amazing, and it's less than 2 seconds. So this is
Okay, wait.
something
I
you
have
can
so many
observe.
questions. Okay.
Okay.
So they don't have like a penis bone or anything. Is it inflatable
Correct.
penis? It's an inflatable penis. Okay.
So it's not vascular like most mammals.
Mhm.
It's
Right.
fibroelastic. So it's full of
Okay.
tough elastin tissues
Hmm.
and very fibrous, very similar to the closest relatives, which are even-toed ungulates like cows and pigs and sheep.
Okay. So when it's not erect, what does it
It's
look like?
it's still inside the body and it wouldn't be completely flaccid ever. So it still
Okay.
is in a semi-turgid state always, but it further engorges when the animal is excited. And it
Cool.
distends quite a bit. So a lot of
Yes.
the research that I started doing was taking these dead animal penises and inflating them with pressurized saline or melted Vaseline to simulate erection and understand more about their shape. So
Mm-hmm.
they can distend quite a bit further even after the animal has died over there.
Wow.
So with these harbor porpoises, we started noticing that they only mate on the female's left-hand side. We had something like 142 mating events, and every single one, the male was on the left-hand side.
Did you think to collect those data at first, like from the very get-go? Did you always collect which side they were on, or was it like happenstance that you started noticing
No,
this?
I was very fortunate that I was a collaborator on a project with my colleague Bill Keener and some folks at the Marine Mammal Center in California in San Francisco. And they've been studying the harbor porpoises there that mate underneath the Golden Gate Bridge. So the porpoises disappeared during the war because they put submarine nets underneath to keep submarines out of the bay. And when they removed the nets, the porpoises started to repopulate the area. And this team of researchers found that if you stand on the Golden Gate Bridge at high tide, you can observe harbor porpoises mating every single day. So
What?
food moves into the bay, females follow the food, the males wait for the females under the bridge because it's the narrowest point and they ambush the females there. And we think it's a stealthy approach. We don't think they're making echolocation clicks, which is also really interesting because usually males alert females to their presence when they want to mate. There's copulatory behaviors. We think the females are caught off guard and they are totally ambushed, which is really unusual as well. There's so much weird stuff going on with harbor porpoises. But the males only mate on the left-hand side. And what it turns out is happening is that is the only way that the penis can bypass all these vaginal folds. And in any other angle of approach, the penis would get caught on one of these vaginal folds and be forced to basically ejaculate far away from the cervix and far away from where there's a possibility of actually inseminating the female. But if he approaches just right, which is actually on her left side, he can penetrate the deepest, spiral through these vaginal folds. Something amazing also is the penis we believe to be prehensile, that males can control them
What?
voluntarily. Someone once sent me a video of a captive dolphin that was trained to move its penis in time to snake charmer music, which
No.
was mind-blowing.
What's
So
that?
if you think about it, you have animals with these very large penises coming at force in less than 2 seconds with a prehensile tip that can navigate through these little blind end recesses and all these spirals in there. The whole process is fascinating. And it's this
Yeah.
amazing level of coevolution of not just anatomy between the sexes, but also behavior. That males are only mating on the left-hand side because that's the only way to bypass these vaginal folds in there. And we have no idea what came first, the chicken or the egg. So did the females have the behavior or the anatomy first? We don't know, but the whole process is an excellent example of an evolutionary arms race to control paternity.
Do females show any kind of resistance behavior?
We have looked at female responses and categorized them, and for the most part, they're pretty mild responses compared to animals with more contest competition where males are physically competing with each other. For the most part, the females might dive down a little bit or kick their tail flukes out, but they're pretty mild responses over there. But what I do think females can do is they can shift their body slightly left or right, which would offset the penis inside that reproductive tract. So if the penis is not perfectly aligned, that means that male is unlikely to fertilize her egg. And so by slight body shifts and this rapid 1 second interaction, a female might be able to control paternity. So it's not just this passive female approach. They might have a very solid mechanism in terms of their anatomy and behavior to control who fertilizes that egg.
It's interesting. And if a male doesn't get just the right position, then the sperm ends up in one of the folds and eventually gets shed. Is that what happened?
So the sperm could get caught underneath one of these flaps and never pass forward. It could be exposed to salt water. It could make its way through. We know that the vagina in general is a very hostile environment towards sperm. There's lots of female mechanisms in terms of the biochemistry, things like even the pH, which are going to affect sperm viability. So from a male's perspective, you want to deposit that sperm as deep as possible, as close to where the egg is as possible, so you're most likely to be able to reach that egg.
And do females mate with multiple males per reproductive bout?
Absolutely. I would not want to be a female dolphin, that's for sure. So it seems that, for example, I do a lot of work on dusky dolphins in New Zealand, and we'll observe 10 males mate with a female over the course of 10 minutes. So it's this constant mating with females over there. They have a very short estrus period, so we know the actual time where a mammal is ovulating and can possibly be impregnated is very, very short. We're talking a couple days at maximum, but they are mating for months at a time as well. So males are trying to time it to be able to know when that female's is in estrus and ovulating, and to be able to fertilize at that exact moment in time.
And do sperm stay alive within the female reproductive tract? Like, is there any short-term sperm storage or anything like that?
Probably not. So
Okay.
dolphins have pretty typical mammalian sperm, which
Mhm.
means that they don't remain viable for a very long
Mhm.
period of time.
Got
So
it.
I know in insects there's a lot of sperm storage organs, and we're
Yeah.
not finding those in our mammals.
In the bird, sometimes you see these sperm storage tubules
Mhm.
also. Yeah.
Exactly. Yeah.
Very interesting. I know that some of your recent research was on clitorises in dolphins. And so I'm curious if you can tell us, like, first, just define how would you even recognize a clitoris in another species? And I imagine it's different if the species is sort of closely related to humans versus not, and how you go about finding the clitoris. Why don't we start there?
I think that's a long-standing question for a lot of people of how to find
Yes.
the clitoris.
How do you find the clitoris, right? Yes.
The whole concept behind the project is that people would come up to me and say, "Oh, you study dolphin mating. I read on a Snapple bottle that dolphins have sex for pleasure." And I realized that no one had actually ever tested this hypothesis before. And the idea is that dolphins are mating all year round, even though their conception period is really short. And it could be, though, for other reasons. It could be social bonding. It could be working out dominance relationships. It could be practice. We see calves mating with their mother often as well. But one thought was that it was for pleasure, but no one had tested it before, which was the
And yet
whole
it was on a
idea.
Snapple bottle. Be
It was
careful where
on a
you get
Snapple.
your information.
Exactly, exactly. So I wanted to test this idea. And this is something that I did with Patricia Brennan, my postdoc advisor.
Uh-huh.
And she knows so much about clitorises. If you think about it, they've only been studied in a couple handfuls of species. We know very little about them, but they exist in theory. And all mammals, all our reptiles, and all our birds should have a clitoris.
Why? What makes you say
Because
they
it's
should?
evolved, like it's a historic trait that is in
Okay.
amniotes. So we should find
Ah.
them in mammals, we should find them in reptiles, we should find them in
Mhm.
birds, but no one's looking for them yet. And there's a lot of
Mhm.
historic stigma I can talk about
Yeah,
why
I'd
there's
love
this
to hear that.
deficit in research.
Yeah.
But they look like kind of like you would expect them to look in a human, in that they are at the opening of the vagina and they are sticking out a little bit as well.
Mhm.
That being said, the clitoris, the part that we see when you see a clitoris, the external part is only a small fraction of the clitoris. The
Right.
name clitoris actually means little mound or little hill,
Mhm.
because the majority of it is deep beneath the surface as well.
You went looking for it in dolphins. Tell us about that story.
We could find them. They were very obviously there, but the
Okay.
question was whether
Yeah.
or not they were functional. And
Ah, okay.
so, are they just there, or do they actually function in some sort of stimulatory capacity?
Mhm.
And so we approached this in a few ways. So we looked at where they were positioned, and we did this anatomically. So we found that the clitoris of dolphins, this is bottlenose dolphins, were really wrinkled. And that wrinkling actually tells us something. If something is wrinkled, it means it has the possibility to expand further when an animal is excited, when it's engorged.
Mhm.
The penis is full of erectile tissue. The only erectile tissue in the female reproductive tract is inside the clitoris.
Okay.
So there's erectile tissue in there.
Yeah.
The animal is excited. It's going to fill with blood. That wrinkling tells us there's a lot of room for engorgement to occur over there. There's a lot of room for that blood flow over there. And then we section down that clitoris. So we first 3D scan them. So we stain them and we soak them in iodine essentially, because when you do a CT scan, a computed tomography scan, they are great at picking up bones and hard tissue, but very hard to pick up soft anatomy. But if you stain them in iodine, and I stain some floors also along the process, if you stain them in iodine for a while, the CT scan can pick them up as well. So this gave us an idea of what the actual shape was, and we were really surprised and excited to find that the shape of the dolphin clitoris is very similar to the human clitoris. It looks pretty much the same.
Wow.
And it also was not, we were thinking maybe there would be some changes based on developmental stage of an animal, if it was a newborn or a young animal or sexually mature or an older animal. And we found the only difference was really just the size. So it's scaling as you would expect, but there was not this big difference between newborns versus a fully developed clitoris. They are similarly structurally as well. And so we started to actually section them and look in different parts of them. And one of the things that caused us a lot of delay on this paper actually was what we found was so surprising and confusing, we were questioning ourselves. And what we found were what we thought were nerve bundles or nerve endings, these nerve fibers that are 10 times bigger than found in a human penis.
What?
So if you're looking at a human penis under a microscope
Yeah.
and looking at sections of these nerves, you might be looking at 100-time magnification. We were looking at 10 times magnification, and they were bigger than you would find in a human.
Wow.
So these huge nerve bundles and more of them also. So that tells us this is a very sensitive region. But we found that we have underneath our skin something called a dermal papilla, which is a very sensitive region, very close to the skin. And that's where these nerve endings are reaching. And we found that they were very close to the end of the skin in the dolphins at the clitoris. So this suggests these nerves are going all the way to the very, very tip of that clitoris skin over there. So collectively, these were telling us that there's a lot of similarities in shape and structure between the dolphin and the human clitoris, and that they have these huge nerves, which are highly suggestive that we have these functional structures. We can't actually say if a dolphin is having an orgasm or not. The way you would look at that in a human might be things like physiology. You'd look at if an animal is breathing heavier or making sounds or curling their toes. Dolphins don't have toes. Very hard to do these other things underwater. So we'd have to do some sort of physiological test to confirm if an orgasm were to occur. But certainly they have the structures suggesting that they are functional clitorises capable of feeling these stimulations.
Do female dolphins masturbate?
There is very little evidence of it. I think people just are waiting for multiple examples to be able to publish on it. But there absolutely are some anecdotal reports of females rubbing against structures. And we've seen males will put their beaks against the female genitalia and echolocate into that.
No.
So you can think about echolocation as a very, very high intensity sound creating
it's a little vibrator.
vibrations.
Yes.
Exactly.
Oh my gosh.
So that might be what's going on. So there is, we think that this echolocation, females will also echolocate on each other's genitalia. So we think that's a form of maybe some sort of foreplay.
Very interesting. Cool. So earlier you were alluding to kind of the history of research on animal clitorises and why it was so understudied, or maybe genitalia, female genitalia in general. I was wondering if you could expand on that.
Sure. So compared to male genitalia, female genitalia has been vastly understudied. And this is a historic trend which has continued even though there's no reason for it to continue. So the historic reasons were that there were more male scientists than female scientists, and males were more interested in penises than they were in vaginas. So that makes intuitive sense. Another logic was that the penis is external, the vagina is internal, so it's easier to study something which is sticking out, which is internal. I can understand that as well. But now we have these amazing CT scans and all kinds of tomography where we can, scanning electron microscopes, where we can see what's going on internally. So that doesn't hold a lot of validity anymore. There also is the idea that vaginas simply weren't exciting. There wasn't diversity, that they all were going to look the exact same, whereas penises have all this amazing
vestibule to
morphological.
receive the penis, right?
Right. Yeah. It's just this vestibule, and there's no diversity there. Well, clearly we're showing there's a lot more diversity than we thought there was, just no one's been looking for it as well. If you think about a penis, like let's take a snake penis, which has two hemipenes on it. It has a double-headed penis. Well, the vagina is going to have double heads as well. Same with our kangaroo. So if you're finding these things in males, it's pretty safe to assume you're going to find the same level of a match in terms of anatomy because they've co-evolved together in those females. So that was one part to it, that it was just assumed that they weren't going to be variable. But then I think the biggest factor just had to do with social stigma and the idea that it was considered a social faux pas to talk about female reproductive anatomy. Even now, if you think about your junk mail and your spam mail, you're getting emails constantly for different things to enhance erections, but you're not getting different spam mail about, say, polycystic fibrosis or other female genital diseases. It's all about how to increase erections and male fertility. There's just not that same level of equality within our society when we consider talking about penises. Even in slang, there's so many words to say penis, but very few to say vagina in comparison. So in the 1990s, this researcher named Pendergast was the first one to characterize vaginal diversity in humans, and she showed that humans have slight differences in vaginas, and she was called a dirty old woman. Like she was socially called some sort of pariah for even looking at these sort of questions.
Perverted, all the things.
Exactly, which is crazy to think about that. There was no need at the time or thought why you would ever look at female vaginal diversity. And I can think of an example in my own life where I was going to do some research on penises and vaginas, and I was doing this in a foreign country. I won't mention which one, but I had talked to a university there and had asked to suspend their penises from their ceilings of a laboratory so I could 3D scan them and digitize them as you do when you're me. And the person I was talking to got got back to me and said, well, we have female students, and I think they would be very offended to look in a window and see a penis hanging there. And I reminded them that if I were studying something like a toe pad of a gecko, probably they wouldn't be objecting, but that we are inherently having this bias against genitalia just because it's genitalia,
so squeamish,
and devaluing the science, even though there's the same validity and same need for rigorous science to find patterns there.
You get that in a lot of ways.
And we're still, I mean, the name for the clitoris, so one of the things that it used to be called was the little hill or the little mound, but another name for it was the shameful member. It was just shameful to talk about female genitalia. And if you think about most organs in our body, there is a National Institute of most organ systems in the US, but not a National Institute for female genitalia that falls under children. So we're just not where we need to be yet. But I think conversations like these are so important to help destigmatize topics like talking about genitalia, using appropriate words as opposed to some sort of euphemism. We need to call anatomy and bring the conversation forward, advance it to get to a stage where we can talk about genitalia. And even now, female genitalia are still understudied compared to male genitalia. So even though we've gone through all these other reasons and there's no rationale now, we still study penises more than vaginas. And interestingly, we say male and female as opposed to female and male.
Oh, well, all the time. Yes.
All the time. Well, I'm very conscious. I don't do it anymore.
I know. I catch myself and I change it. So one of my students was wondering how knowledge from dolphin research on female genitalia could be applied to understanding humans or other species or for conservation. Are there some ways that it can be applied in that way?
Absolutely. And that's exactly what we're doing in my lab and something I'm very excited about.
Oh, cool.
We've been using dead dolphins and casting their vaginal shapes because cetaceans have these super complicated reproductive tracts, so they're a good model system. We cast their vaginas and we use those to 3D print and then to digitize and do all these crazy different things with silicone, and we make these bio-inspired artificial vaginas, which simulate the natural shape and elasticity of a real dolphin vagina. And these are being used by trainers to try to improve ejaculate quality. If you use something which feels like a vagina, do you improve ejaculate? And the answer so far seems to be yes. And this is a game changer when you think about it, because we have lots of animals which are endangered, and there are programs where they're trying to breed them and repopulate them in the wild. And if we can actually find a way to increase the success of artificial insemination, we have a way to help with conservation, to help a lot of species. And so a lot of research on artificial insemination focuses on how do you best preserve the semen and preserve the sperm once you've collected it? How do you cryopreserve it? How do you thaw it? What process is how do you actually inseminate it? But there's very little research in the past several decades on how do you improve the quality at the time of collection. So if you're starting with a better quality sperm to begin with, you're increasing your likelihood that that sperm is going to actually fertilize an egg. So we're super excited about where the potential for this research is. It could even go towards things like the agriculture. Think about cows and pigs and all these animals that we're consuming. If we can find a way to produce faster and more rapid breeding of these animals, this has a lot of value towards society.
Right. And all sorts of species, this artificial insemination is used for conservation breeding. We actually just talked to another person, Meg Martin, about conservation breeding, but we didn't talk about the ejaculate aspects of it. So it really ties in. You can imagine doing that for a lot of different species if you can train the males to use these artificial.
Dolphins are very happy to ejaculate and to be trained to ejaculate. That's apparently they're easier to train than most species.
How the heck do you do that? Do you know?
It's a gradual. I have not personally trained them, but I'm told that it's relatively easy to other animals, and it's a gradual process where the male will have an erection on his own volition, and you positively reinforce that, and then you put objects touching the penis until it becomes associated that there's a command to have that erection. So all these dolphins that we're working with at many facilities are voluntarily providing the semen. They're not being anesthetized. Yeah,
they're not being shocked, electro ejaculated or anything like that. That is super cool.
Yeah, we're really excited about where we can go with this research. So it's exciting times ahead.
Yeah, absolutely. What other future research are you excited about?
We are currently working on some projects trying to look at different human contaminants inside sperm. And that's something that we don't think about
sperm?
A lot.
Okay.
So we've done, and by we, I mean my PhD student, the royal we, has done an amazing job with this, but we understand a lot about hormones inside sperm. And we know a little bit about some contaminants, but no one's ever looked for pharmaceuticals inside sperm before. So we know there's microplastics possibly in sperm because microplastics are everywhere in the world. But what exactly is entering our body and is landing up inside our sperm? And that's something we're trying to understand now. And recent advances in mass spectrometry have really made it possible to do these various essentially assays and look at thousands of chemicals and compounds at once and look for matches in there. So we have been working very diligently on figuring out what weird hormones that you wouldn't expect to find and what human pharmaceuticals and pesticides and other pollutants are there. And it all started
specifically in dolphin sperm, right? Not
right.
human
I'm
sperm.
working with dolphin sperm, but
Okay.
this could be expanded.
Sure.
Have the protocol, we could expand this to any species.
Yeah.
And it started because I was having a talk with a friend of mine while we were walking on the beach, and she said, "Is the reason people are tired after sex because of all the melatonin in sperm?" And I said, "What are you talking about? I had no idea there was melatonin in human sperm." And this got me thinking, well, what other hormones are in sperm that you wouldn't think would be there? And what are their functions?
Is it in sperm or is it in the seminal fluid?
I believe it's in the seminal fluid.
Seminal fluid.
I believe it's in the semen. So
in the semen. Yeah.
What we're looking at now is we're actually looking at samples. We're separating the sperm from the seminal plasma and looking at all these different combinations to figure out where these different compounds are found. Is it in the sperm or is it inside the seminal plasma?
Gotcha.
But if you think about hormones, you wouldn't expect to find melatonin intuitively inside sperm. So now we're looking at a whole suite of really unusual hormones and seeing what we're finding there.
How do you take a vaginal swab on a dolphin?
It's not as hard as I've seen videos. I'm the researcher just sending out the protocol and sending the supplies and sending the money. But I've watched the videos and the females are trained to invert their bodies so that their bellies are near the surface. And the trainers will dry the surface area and then stick what looks essentially like a cotton swab that's very long inside the female and swirl it around and then pull it out, cut that off, and then we stick it in a solution which is going to preserve all the DNA in there. So I'm having a lot of fun with the research that we're working on these days. There really seems to be a sky's the limit possibility because there's so much which simply has not been done. And especially in our larger animals, a lot less has been done compared to some of our smaller lab-based animals.
When I was talking to David Hoskin, he was talking about how little had been done on females. And I think he was talking about his own choices to study male genitalia rather than female for the most part, and said, this is going to sound like the ultimate cop-out, but it was just hard, right? To study females. But that's what you're saying. Like, yeah, it's hard, but it's easier now. And it's not that hard, that it's very doable. And
I've had male collaborators say to me, like, you can do it because you're a young female, but if me, an old creepy male, were to do this, or me, not an old creepy male, but if an old male were to do this, there might be more stigma. You can do things because you're young and female. And I don't know if that's true, but it certainly is no shortage of questions, no shortage of opportunities. Most animals out there, we know very little about their vaginal morphology, about their clitoris morphology, about their cellular structure, molecules. There's just so many. We didn't even know what the pH of a dolphin vagina was before my PhD students started working on these things. Like some things which seem to be so intuitive, that dolphins have sex for pleasure. Do they have sex for pleasure? These are all the sort of questions that we're trying to answer now, these basic fundamental questions. And it really gets me thinking about how much we take for granted or assume that we know everything there is to know about anatomy, and we are missing a lot of the basics.
Yeah. And it's interesting. I find people are more comfortable talking about the sex lives and the sexual anatomy, reproductive anatomy of other animals than humans. So it always makes me think, oh, we should just do stealthy sex ed in places that are prohibiting accurate sex ed in schools. Like, yes, go to schools and talk about the clitorises of dolphins or whatever, like, and then maybe people will learn that, yes, humans also have these structures. So I think there's a lot of opportunity there too.
And I think people are also, the world is changing, and I think people are becoming more open-minded about the importance of these research topics. So I've been asked to give talks to school groups, and I've told them what the topic is, and they've said, do it, go for it. Our students are mature enough, even though they're only in high school. But these are conversations and things they need to learn about. So please don't hold back on explaining the anatomy.
That's awesome.
And that's really, and these are including in very conservative states. I've been asked to give talks like these as well, which is encouraging.
Well, Dara, this has been a fascinating conversation. Thank you so much for joining us.
It is my absolute pleasure. I've really enjoyed chatting with you.
Great. We'll talk again soon. And thank you all for listening. Thanks also to our production team, Isabel Espinoza, Lane Patton, and Daphne Trilliana. The music used in this episode is from I Don't Smoke by the Mythical Score Society.